Today, we will be presenting on mutations in Tetrahymena. Before we get to the details of the experiment, we need to briefly talk about some key concepts. Two structures that aid in the movement of cells or organisms are cilia and flagella. Flagella are long hair-like projections from a cell surface. Cilia, on the other hand, are shorter hair-like structures that are located on the surface of many organisms or cells. There are few flagella per cell, while cilia are present in large numbers. They are important for aiding an organism's movement, particularly swimming. Flagella move by rotating in a clockwise or counterclockwise directions, similar to the way a propeller moves. There are two different types of cilia. The first type is motile cilia, which are important for movement. The motile cilia move in a rhythmic fashion. The second type of cilia is sensory cilia. Sensory cilia move in an angular rotation direction and important for signaling. There are two strokes to ciliary movement. The first is the power stroke, and the second, there is a recovery stroke. During the power stroke, the cilia move forward against the surrounding environment, propelling it forward. The recovery strokes allows the cilia to return to its normal position.
Cilia are made of a microtubular backbone called the ciliary axoneme, which is surrounded by a plasma membrane. The axoneme consists of 9 pairs of microtubules that form an outer ring and 2 central microtubules. Motor proteins called dyneins are located between the 9 pairs of microtubules that comprise the outer ring. In the cross-section of the cilia, you can see the dyneins that are connected to the microtubules. These dyneins help facilitate the movement of the microtubules. The arms of the dyneins sliding on the microtubules lead to the bending, causing the cilia and flagella to move. The dyneins are made out of several chains, including heavy, intermediate, light intermediate, and light chains. These chains help regulate the function of the dyneins. There are quite a few genes that code for the different light chains within the dyneins, one of them being the LC4B gene. The LC4B gene is a light chain subunit of a ciliary 3-headed outer arm dynein. This is a protein-coding gene.
Our goal for the experiment was to mutate the LC4B gene in Tetrahymena. To do this, we isolated the DNA from Tetrahymena, cloned the DNA, then ligated the gene of interest with the plasmid, and then transformed E. coli cells with the ligated plasmids. We then disrupted the gene by inserting a DNA sequence into it. After this, we inserted the disrupted gene into the Tetrahymena.
So, why Tetrahymena? We used Tetrahymena for our experiment because they are ciliate model organisms. Because of this, they are ideal for the study of cilia and their function. They are considered model organisms for many reasons. They are ideal because they are large in size, can grow fast, are unicellular, their genetic markers have been thoroughly mapped, and they have well-developed forward and reverse genetic tools. The forward genetic tools aid in the mutation of Tetrahymena. This is especially important to our experiment because our goal for this experiment was to disrupt a gene and create a mutant Tetrahymena.
The first part of our experiment was isolating the DNA. We isolated the DNA by lysing Tetrahymena cells using a urea buffer and extracting the lysate with phenylchloroform isoamyl alcohol. The phenylchloroform isoamyl alcohol is important for separating the aqueous and organic phases from each other. When the phenylchloroform isoamyl alcohol was added, we were able to wash the lysate by extracting the organic phase containing proteins from the lysate. After washing the lysate, we washed the DNA with ethanol. By doing this step, we were able to get clean, isolated DNA. Having the clean, isolated DNA ensures that our sample is free from as much cellular debris, such as protein, as possible. DNA accounts for a small percentage of a cell's material and is concentrated in one spot on the cell. In eukaryotes, this is the nucleus. The quantity, quality, and integrity of the isolated DNA has a big effect on the experiment's outcome, so it is important that we correctly isolate the DNA from the nucleus and make sure the sample is free from other cellular debris. In our experiment, the DNA isolation did not properly work, so we used DNA that was provided to us by the instructor. DNA isolation is a key step because it is required for the polymerase chain reactions, also known as PCR, gel electrophoresis, and cloning.
The second part of our experiment was cloning the gene of interest. In order to increase the quantity of DNA, we used two techniques. The first technique in the process of cloning was a polymerase chain reaction, also known as a PCR. The second technique was inserting the product of the PCR into vectors and transforming them with bacterial cells. For the first technique, a PCR was performed in order to amplify our gene of interest. Amplification occurs when there is replication or an increase in the number of copies of a gene of interest without increasing the number of copies of the other genes. In this case, the gene of interest is the LC4B gene. PCR essentially finds the DNA or specific gene of interest and then amplifies it using DNA polymerase. We designed primers that are complements to the ends of the gene of interest to bind to our gene of interest in order to guide replication of our gene. The PCR then repeats the cycle of DNA synthesis, which leads to the synthesis of many copies of the LC4B gene. After running the PCR, we ran an agarose gel electrophoresis to ensure we amplified the correct gene. Once we knew we had amplified the correct gene, we ligated our gene of interest with pGem-TEZ plasmid. This particular plasmid acted as a vector for cloning. This was the second part of cloning. Inserting the gene of interest into a vector or plasmid creates recombinant DNA. We did this by essentially letting the pGem-TEZ vector and our gene of interest hang out in an Eppendorf tube with ligase. This allowed us to insert our gene of interest into the vector. Once the gene was inserted, we transformed the plasmid containing our gene into E. coli cells. This process takes advantage of the genetic process of bacterial cells. Amplification of the plasmid occurs in the same way the replication of the bacterial chromosome would replicate. The E. coli cells we used had been modified so they did not contain their own plasmid. This allowed us to transform our plasmid into cells.
We did this by creating holes in the E. coli for the plasmid to enter and sealing the holes back up by changing the temperatures. By subjecting the E. coli cells to high temperatures, we were able to create holes in the cell's membrane and compel the cells to take up our gene. After the gene was taken up by E. coli, we exposed the E. coli to a lower temperature to seal the holes created by the high temperature. This created E. coli cells that contained our gene of interest, which allowed for a system of more and more gene of interest to be formed. After the cells were transformed with the plasmid containing the gene of interest, the E. coli cells were plated on an LB plus AMP plate. We did this because the vector we used, pGem-TEZ, contained an LB AMP resistant gene. When the bacteria were plated on agar that contained antibiotics, the E. coli cells with the plasmids containing the resistant gene grew, and the cells that did not take up the plasmid did not grow. This created a selective advantage for the transformation of E. coli cells to grow and replicate.
For gene disruption, we used the plasmids containing our gene of interest. We chopped up the central region on the gene of interest, which created a gap. We then inserted a new DNA sequence in the gap that was made. The new DNA we used was a neomycin resistance gene. We used this specific neomycin resistance gene because it produces a protein against neomycin. We used this specific neomycin resistance gene because it produces protein against neomycin. This is an important factor because when testing to see if the tetrahymena contains the disrupted gene, it is placed in a media containing neomycin. Only the tetrahymena containing the disrupted gene will survive because of the resistant gene they now have. This insertion of a new DNA sequence into the gene of interest disrupts it, rendering it non-functional.
The final part of our experiment was inserting the disrupted LC4B gene into Tetrahymena. We were unable to complete this step at Simpson's labs due to lack of technology, so we sent our plasmid containing the disrupted gene to Cornell College's Stock Center to be inserted in Tetrahymena. To insert our gene of interest into Tetrahymena, it first has to be isolated and cut out of the plasmid. This way, only the LC4B gene containing the neomycin resistance gene is left and is ready to be inserted into the Tetrahymena. The DNA segment is then shot into Tetrahymena using a gene gun. Then, through homologous recombination, the new DNA becomes part of the Tetrahymena. Homologous recombination is when genetic information is exchanged between two molecules and it occurs naturally during mitosis.
So, to conclude, after inserting the mutated gene back into Tetrahymena, the replication of the gene will take place. It will be transcribed and translated into proteins, and polypeptide chains will form. However, these polypeptide chains will not make sense and will be unable to be read by the cell because these proteins are now irrelevant to the cell since they are mutated. The LC4B gene codes for proteins in the outer arm light chain dyneins. A disrupted LC4B gene will cause a good percentage of the light chain dyneins to not be assembled because the accessory proteins will not be integrated. This makes the dyneins unable to move along the microtubules and causes dysfunction in the way cilia beat. Cellular biological analysis can investigate further into the dysfunction of cilia. Through this analysis, experiments can be done that focus on wave formation of cilia, frequency of beats, and swimming speed. This entire project incorporates everything we have learned in class, including topics such as transcription, translation, and gene expression. Without knowing these processes, seeing the effect of the mutation in Tetrahymena would not be possible. It is important to understand the processes in mutating the LC4B gene in order to understand how the cilia function in Tetrahymena and how we can apply this knowledge to other areas of interest.
Cilia are found in almost every cell in the human body and perform a variety of functions that enable the body to maintain homeostasis. Tetrahymena are frequently used in research for ciliary human disease because their expressions of mutation is very quick and lethal mutations can be studied easily to determine the mechanism of the mutation that leads to the cell death. Primarily, cilia are especially important in ciliary signaling. If cilia malfunction, cell signaling is affected and can lead to a variety of human diseases. One example of improper signaling of cilia occurs in kidney disease. A malfunction in the signaling causes fluid to build up in the kidneys, which leads to the formation of cysts that can clog the channels in the kidneys and prevent blood purification. These studies using model organisms like Tetrahymena give scientists insight into what the functions of different proteins in cilia are. This can help us understand more about human disease by looking at what is wrong with the cilia and tracing it back to what protein and gene might be the problem.